Hey everyone, and welcome to this explainer. I am so thrilled to have you here today. If you've ever stopped and wondered exactly how modern medicine takes that massive, terrifying leap from a beaker in a laboratory into a living, breathing human being, well, you're in the right place. Today we're going to explore the ultimate gatekeeper of medical science, Phase 1 clinical trials. We'll be breaking down exactly what happens when a brand new treatment meets the human body for the very first time.
So here's the question to get us started. How do we know a new drug is actually safe before it reaches the pharmacy shelves? I mean, we can't exactly just synthesize a new chemical, test it in a lab, and then start handing it out to the public, right? There are literally immense life or death stakes involved in moving a new, unproven chemical from animal testing into the human body. It requires an incredibly careful, highly regulated environment to make absolutely sure things go right.
Here's our roadmap for today's explainer. First, a welcome to Phase 1. Second, before human testing begins. Third, finding the limit, or MTD. Fourth, choosing the right subjects. And finally, limitations and next steps.
All right, let's jump right into Section 1. Welcome to Phase 1.
Phase 1 is absolutely crucial. It's the literal bridge between the research lab and real-world clinical application. This is the very first stage of testing in humans. As our source material beautifully puts it, Phase 1 serves as an interface between preclinical research and clinical drug development. What this means in plain English is that we're actively taking data from animal models and using it to make reliable, rapid predictions about human responses. It's an interface. It's a careful translation of preclinical toxicity and safety data into the incredibly complex realm of first human exposure.
So what are we actually measuring? Well, one of the primary objectives of Phase 1 is understanding pharmacokinetics, or PK for short. You can just think of PK as what your body does to the drug. When a person takes a dose, scientists need to measure exactly how it's absorbed, how it distributes through the tissues, how the liver and other organs metabolize it, and finally, how it gets excreted. Safety and tolerability are the main goals here, and PK is the engine that drives that understanding.
Then, as a secondary objective, researchers also measure pharmacodynamics, or PD. If PK is what your body does to the drug, pharmacodynamics is what the drug actually does to you. Scientists are looking for the biological and physiological effects. Between PK and PD, researchers can paint a really complete picture of how this chemical behaves in a living human system.
Now you might be wondering how many people are actually taking these early risks. Honestly, it's usually just 20 to 25 healthy volunteers. It's an incredibly small, intimate group. A typical Phase 1 study runs for about 6 to 12 months, and importantly, it's usually open-labeled. That means there's no blinding involved. Both the doctors and the patients know exactly what drug is being administered. This is absolutely vital for monitoring immediate safety. But obviously, you don't just grab 20 people off the street and start handing out a new chemical.
Which brings us to Section 2, before human testing begins.
There are incredibly strict regulatory hurdles and unskippable prerequisites that have to be cleared before anyone takes a single dose. First up, scientists must have comprehensive preclinical data. Second, they have to file an investigational new drug or IND application and secure official regulatory approval. Third, there must be strict informed consent from the participants, and this requires absolute adherence to the Declaration of Helsinki and ICH-GCP guidelines, not just at the start, but continuously monitored throughout the whole study. Patient safety is locked in from day one. Only once all those approvals are in place can the trial actually begin.
Okay, moving to Section 3, finding the limit, or the MTD.
The primary aim of these early trials is an extremely delicate balancing act of biology. They're trying to find the maximum tolerated dose, the MTD. Think of this as finding the safe ceiling for a drug. To find the MTD, researchers systematically escalate the treatment dose until they hit what's called dose-limiting toxicity, basically the exact point where adverse toxic effects become apparent. By finding that ceiling, they know exactly what dosage ranges are safe to test later on in development. Looking at the comparison table here, we can see exactly how they find that ceiling. There are two main approaches, SAD and MAD. In SAD, or single ascending dose studies, tiny groups of 3 subjects are given a single dose and observed. If there are no adverse effects, the dose is bumped up and given to 3 new healthy subjects. But if there is toxicity, 3 more subjects are given that exact same dose just to confirm. If it stays toxic, boom, that dose is officially considered the MTD. On the flip side, we have MAD, or multiple ascending dose studies. These are conducted to understand the PK and PD of multiple doses. A group receives multiple low doses, and researchers collect blood and fluid samples over time to analyze how the drug is processed. Oh, and as a quick aside, they'll also sometimes do food effect studies to see how eating a meal changes the drug's absorption. Kind of crazy to think about, right?
That brings us to Section 4, choosing the right subjects.
So we've talked about what the subjects do, but who exactly are they? As you can see, when comparing volunteer types, researchers normally rely on healthy volunteers. And why is that? Well, they offer a large recruitment pool, they can be recruited super fast, they form a more uniform group, and they show greater compliance with the testing rules. Crucially, their results aren't messed up by an underlying disease. And, heaven forbid, if there is an adverse reaction, healthy people just have much better chances of a speedy recovery. The practical advantages here far outweigh the disadvantages, even though these healthy subjects get zero therapeutic benefit from the drug. However, sometimes patient volunteers absolutely must be used. This happens whenever preclinical toxicity data flags a massive potential risk for a healthy person. For example, highly toxic cancer treatments, AIDS therapies, or advanced psychiatric drugs are tested directly on patient volunteers in advanced stages of disease. Testing those harsh dose ranges on healthy people poses way too great a safety risk and violates those strict regulatory guidelines we talked about earlier.
Regulators also step in and mandate the inclusion of special populations to ensure safety across different demographics. We're talking about women of childbearing age, children, but only if the new chemical entity is specifically proposed for pediatric use, of course, and the elderly, meaning anyone over 65 years of age. Understanding how these specific diverse groups process a drug is a vital requirement before moving the drug forward.
And finally, Section 5, limitations and next steps.
When we look at the inherent drawbacks of Phase 1, we hit a fundamental tension. Trials are restricted to highly homogeneous subjects. Like we discussed, these are small, highly controlled, uniform groups of just 20 to 25 people. Yet the performance of this drug is ultimately being extrapolated out to a massive, wildly diverse, real-world marketplace. You're trying to predict how millions of vastly different human bodies will react based on data gathered from just a couple dozen carefully selected individuals. That's a huge leap, right? And that's exactly why Phase 1 is just the beginning. It paves the way for the much larger, much more diverse Phase 2 and Phase 3 trials.
Phase 1 trials truly are the ultimate gatekeeper. They balance this urgent, desperate need for new medical breakthroughs with the absolute necessity of rigorous, uncompromising human safety. But it leaves us with a pretty fascinating question to ponder. If this much scrutiny is required just to test a drug on 20 people, how monumentally complex do you think it gets when rolling it out to 20 million? I really hope you found this breakdown of Phase 1 clinical trials insightful. Please like, share, and subscribe our channel to keep learning with us. See you next time.